All right. All right. Welcome back, everybody. Welcome to this next session, very special session, Tissue Chips in Space, exploring therapeutic drug development and countermeasures. We have with us some very distinguished researchers who have been doing incredibly innovative and important research in that area. Leading the panel today is Dr. Michael Roberts. Mike is a vice president at CASIS and acting chief scientist of the International Space Station U.S. National Laboratory. Mike, please take it away.
Thank you, Jim. I also want to extend a thank you to the National Center for Advancing Translational Sciences at the NIH for their collaboration with the ISS National Lab in funding the Tissue Chips in Space initiative and taking the lead for our nation and helping to continue to fund and advance this important technology. Together, we aim to accelerate translational research and tissue chip technology on the ISS to expedite therapeutic development that impacts humans here on Earth. The panelists in this session this morning were all supported through this collaboration between NCATS, NIBIB, and the National Lab. And with that, we'll go to our first presentation. Dr. Duck-Ho Kim is an associate professor of biomedical engineering at Johns Hopkins School of Medicine. Duck-Ho's research spans the boundaries between nanotechnology, biomaterials, and mechanobiology, with an emphasis on applications for tissue engineering and regenerative medicine. Dr. Kim is also a founder of NanoSurface Biomedical, now Curie Bio, which is focused on integrating human-induced pluripotent stem cell-derived cells, tissue-specific biosystems, and artificial intelligence-enabled data analytics to accelerate the discovery of new therapeutics. Recently, Dr. Kim and his team sent an engineered heart tissue platform to the International Space Station. To tell us about the work in his lab this morning, we will hear from Devin Mayer, a doctoral candidate in Dr. Kim's lab. Duck-Ho will join us later in the session for questions and answers. Let's begin.
Hello, everyone. My name is Devin Mayer, and I'm a doctoral candidate in the Department of Biomedical Engineering at Johns Hopkins University. Today, I'll be giving an update on our engineered heart tissues in space project. As you likely know, spaceflight affects almost every physiological system in the human body, including the cardiovascular system. NASA has identified several cardiovascular risks as areas of high priority for research, including diminished function, occurrence of serious cardiac dysrhythmias, and impaired responses to stress and exercise. While we've gained important insights from prior studies, a shift to using 3D and vitro models of the human myocardium could improve our understanding of microgravity's direct effects on the human heart. Furthermore, findings from these studies on microgravity may help inform research on the effects of aging here on Earth. The main goal of our project is to develop a high-throughput microphysiological model of the human myocardium derived from human-induced pluripotent stem cells to study the effects of microgravity on cardiac tissue structure and function with the goal of developing mitigation strategies. We've been working on this project over the past 3 years, sponsored by the NIH and CASIS. In this time, we've designed and validated our engineered heart tissue, or EHT model. In 2020, we were able to send these EHTs to the International Space Station for 1 month, following which we analyzed contractile function and changes in structure, biochemistry, and genomics. Since this time, we've continued to analyze the return tissues, as well as started to test and study the mitigation strategies used for the second spaceflight mission next year. The engineered heart tissue, or EHT platform used in our study is comprised of induced pluripotent stem cell-derived cardiomyocytes and stromal cells that are seated within a hydrogel scaffold and cast around 2 silicone posts, in which deflection of the flexible post allows us to measure contractile forces generated by the tissues. You can see the deflection of the posts here in this video, and using this deflection, we're able to use these equations to calculate the actual forces of these tissues. Since one of the major roadblocks to using human-induced pluripotent stem cell-derived cardiac cells and tissues for in vitro study is their phenotypic immaturity post-differentiation, we've utilized a novel electroconductive hybrid hydrogel to serve as scaffolds to improve the maturity of engineered heart tissues. This hydrogel is comprised of decellularized extracellular matrix, or DECM, derived from porcine left ventricular myocardium, and reduced graphene oxide, or RGO. By combining the tissue specificity of DCM with the electroconductivity and stiffness of RGO, we were able to generate tissues with enhanced maturity, as indicated by increased twitch forces, upregulation of key cardiac genes, and improved electrophysiological function. This was published within the past year. We further modified the AHT platform by incorporating magnets into the flexible posts. These magnets, when coupled with an array of giant magnetoresistive or GMR sensors, allow for the measurement of post deflection in an automated and real-time manner without the use of imaging modalities. As posts are deflected, the resulting change in the magnetic field is detected by the sensors, and this can be converted into voltage, which is correlated with force, and we're able to use this to calculate force from the deflection of the posts without the use of imaging modalities, as I mentioned. By combining the direct sensing circuit, shown in yellow, as well as reference sensing circuit, shown in red, on this board here, we're able to subtract out noisy signals to give a clean readout. This is especially important as astronaut time on the International Space Station is extremely limited. So we were able, by incorporating these, we were able to minimize the amount of astronaut oversight that we required for our project. Engineered hard tissue arrays were then incorporated into spaceflight chambers that were designed by Bioserve Space Technologies. These chambers consist of a hard polymer body, gas-permeable PDMS membrane, and a self-sealing port for fluid exchanges. Arrays of 6 tissues can be inserted and the whole chamber sealed to prevent leakage, with the end result looking like this. This is necessary as normal cell culture hardware would not function in microgravity. These chambers are then integrated into Bioserve's plate habitat, or PHAB. For this experiment, each PHAB holds 4 chambers, with each chamber pair residing over a printed circuit board containing the magnetic sensors. Once chambers are inserted and secured, the entire assembly is sealed in an anodized aluminum case that contains vents to allow for gas exchange when the PHAB is situated in the incubator on the ISS. This is what an integrated PHAB looks like, and this is postdoc Jonathan Sweep putting the tissue chambers into the PHAB prior to putting it into one of the aluminum cases seen in this picture here. This was all taken down at Kennedy Space Center for final integration and right before the final launch. Here's our experimental design and timeline. Upon arrival at the International Space Station, all tissues were fed or had a media change, and then subsets of samples were terminated at day 7, 14, and 28. Termination consisted of fixation in paraformaldehyde for transmission electron microscopy or immunostaining processing, or preservation in RNA later for RNA-seq. A subset of tissues were also returned viable and cultured for an additional week on Earth to see if there were any recovery effects. With the AHT platform and spaceflight hardware developed, we conducted experiment validation testing, or EVT, which was an assessment of the entire system and all of the procedures we would use for this experiment. This assessment included long-term tissue viability testing, validation of the electronics and software used, testing of the integrity of our chambers during freezing with RNA later or paraformaldehyde, as well as the testing the quality of the resulting immunostaining and collected RNA. We'd initially planned to ship fabricated tissues to Kennedy Space Center. However, our ship test showed that there was an unacceptably high failure rate with regards to tissue integrity. So we instead opted to hand transport our tissues to Florida in these chambers here. So we placed the tissue chambers into this foam cutout in order to secure them. We placed them into these metal boxes that have an airtight seal. These were sealed inside of the incubator in order to capture as much CO2 as possible. These were then sealed further and placed and surrounded with phase change packs in a styrofoam container, and then all wrapped up into our final box here. With the phase change packs holding 37 degrees and the CO2 we were able to capture in these sealed boxes, we were able to transport them for up to 8 hours without any deleterious effects on the tissues. Once our AHTs had arrived at Kennedy Space Center, they were maintained throughout the pre-launch period, after which final tissue quality checks, chamber integration, and hardware and software checks were conducted prior to handover for the launch. On March 5th of 2020, our experiment was handed over and securely packed away in the Merlin unit, seen here, which is a powered incubator that would keep our tissues warm while they made the trip to space. In this picture, you can see one of our PHABs being strapped in, and the entire Merlin unit would eventually be bolted into the SpaceX Dragon 1 capsule. Finally, shortly before midnight on the 6th of March, we were able to watch our experiment get launched aboard SpaceX's CRS-20 resupply mission. This video doesn't really do it justice, but it was an incredible experience, and it really made all of the months of hard work that led up to this worth it. This is a picture of astronaut Jessica Mayer during an engineered heart tissue chamber media exchange, as well as fixation and preservation swap in the Life Sciences glove box in the Japanese experiment module aboard the International Space Station. You can see here the sealable ports in our tissue chambers inside of the PHAB. We're able to stick a syringe in one side as well as in the other, allowing us to push media in through one end and pull the old media out through the other end. You can also see a top-down view of this going on here. Interestingly, Jessica Mayer, the astronaut shown here, was a co-investigator, our co-investigator, Peter Lee and Jessica Mayer, worked together in their final undergraduate year at Brown University. We were excited to find that the EHT survived launch, and they produced contractile function data during their month in space. While the analysis of this data, once the analysis of this data was conducted, we found that there were functional defects in microgravity after just 4 days in microgravity, with twitch forces decreasing consistently over time during their stay on board the International Space Station. Differences in the mean change in beat interval, which is used as a metric for arrhythmia, show a peak at day 19, but a reduction by day 26 and recovery to baseline upon return to 1G. This exciting data shows and recapitulates a lot of the previous findings that show that arrhythmias and decreased contractile function occur in microgravity. Once we received the fixed samples, we performed transmission electron microscopy. While performing this microscopy, we noticed these very large intracellular lipid accumulations quite frequently in our late spaceflight samples, but not in our early spaceflight or ground control samples. This is indicative of oxidative stress, unfolded protein response overload, integrated stress response, and/or mitochondrial dysfunction. This is consistent with previously published work that show that mitochondria serve as a central hub for the effects of spaceflight. Lipid disorders and cardiovascular risk are linked, and there are links between mitochondrial dysfunction and poor cardiac health. Overall, this exciting imaging experiments have shown us one possible cause of the functional changes we saw on board the International Space Station. Now that we know what some of the effects of spaceflight are, we are currently testing therapies. To test these therapies effectively, we needed to simulate microgravity. To do this, we have been using a random positioning machine, which simulates microgravity by rotating the samples in a random pattern, effectively decreasing the gravity force vectors sensed by the tissues to the level of gravity found on the International Space Station. Our spaceflight hardware is placed in the random positioning machine, as shown in this picture here, for the duration of the experiment. In the random positioning machine, we are investigating, in addition to small molecule and drug therapeutics, increases in preload and/or afterload for their potential to prevent maladaptation to microgravity. For increased preload, we've developed a belt-driven actuation system that periodically drives the magnet closer and further away from our post system, shown in this picture here. Bringing the magnet closer stretches the tissue, and this is matched with the relaxation phase of the EHT contraction, mimicking preload. Increased afterload is implemented by using afterload bracers, as shown in this picture here. The way these work, we're able to effectively shorten the bendable portion of the post as these bracers increase the resistance to contraction essentially to infinite levels compared to the stiffness of the flexible posts themselves. What this functionally ends up doing is increasing the stiffness by decreasing the effective height of these posts, and this increases the resistance to contraction, similar to increased afterload. These methods would be similar to an exercise regimen for decreasing skeletal muscle atrophy by increasing the work the AHT must exert in order to contract. However, our spaceflight chambers, as well as our post system for Earth-based studies, are all fabricated using PDMS, which is a polymer very commonly used in the field. Previous work by other groups have shown that PDMS significantly absorbs drugs. So in order to have effective and accurate drug screening studies, we started working on developing methods to prevent absorption by PDMS. In light of this, we were able to find previously published work that showed that adding a PDMS PEG block copolymer decreases the hydrophobicity of PDMS. As the hydrophobic interactions between drug and polymer are one of the primary causes of drug absorption, we integrated 1 to 2% of this PDMS PEG block copolymer into our standard PDMS and assessed drug absorption using UV-Vis. Increasing the PDMS PEG concentration was sufficient to decrease absorption of drug for verapamil, but not Bepredil. We next tested pretreatments in which we would incubate the PDMS with either experimental levels of drug, which was the 100 micromolar, or up to 10 times the experimental levels of drug for 24 hours. By pretreating, we were able to further decrease the amount of drug absorbed in our verapamil samples. This work is currently ongoing, and we are planning to submit this for publication within the next month or so. So in summary, we were able to use a novel electroconductive hybrid hydrogel to improve the maturity of our engineered heart tissues, and by modifying the platform to not only enable automated force sensing, but to also be packaged for spaceflight, we successfully delivered the experiment to the ISS last year. Analysis of the spaceflight contractile data showed functional deficits of our engineered heart tissues. Our ground-based studies have shown some of the structural changes include intracellular lipid accumulations indicative of potential mitochondrial dysfunction. All of these studies are currently ongoing, and we hope to have this paper for the first spaceflight mission submitted within the next couple of months. Finally, we are actively developing and testing strategies for preventing the negative effects of spaceflight on our tissues. This is our team here at Johns Hopkins University, University of Washington, and Brown University, as well as our partners at Bioserve Space Technologies. And finally, we'd like to thank our funding sources, and we'd like to thank you for your time, and please reach out to us if you have any questions. Thank you.
Thank you, Devin Mayer, and everyone in Doug Ho Kim's lab for that excellent presentation. Our next invited panelist is Dr. Siobhan Mullaney. She is an associate professor in the College of Pharmacy at the University of Florida. Siobhan's laboratory seeks to improve in vitro cell-based systems that can predict human drug efficacy, particularly for age-related diseases. Dr. Mullaney is also the president and CEO of MicroGRX, a company that designs and manufactures custom diagnostic microfluidic systems for human research, supporting diagnostic tests here on Earth and in space. Siobhan leads a team that recently sent a human muscle cell tissue chip payload to the ISS, and we are excited to hear more about her progress as we listen now to her presentation.
Hello, my name is Siobhan Mullaney. I'm founder of MicroGRX, and I'm a faculty member in the College of Pharmacy at the University of Florida. I'm going to talk to you today on our panel session on NPS systems on muscle tissue chip autonomous laboratories in space from the past decade to the present. There's certainly been an increase in payloads to the ISS in the last decade, and particularly in technology development and demonstration, biology and biotechnology, and educational activities. The number of payloads has significantly increased, and NASA's payload increase has been nearly 300%. The drivers being cases financial support for research on the ISS, access to the ISS, and the growth of implementation partners that can support these payloads. I came to Florida in 2011, and one of the first things I was able to participate in is to see Endeavour's final flight. And this marked the time where I had conversations about the possibility of sending life science experiments to the space station. And since then, I had been looking for opportunities to do just that. And our first opportunity came in 2015 with funding from Space Florida through the Florida Israel Innovation Program. And this was to develop a small 2U system with Space Pharma to integrate optics, fluid handling, end-to-end experimentation into this small shoebox payload. We sent just 2 chips. This was 2-dimensional primary human muscle cells, so really a cell culturing proof of concept. We received matching funds through CASE's Organ on a Chip Challenge in 2016 to then fly this experiment. And we used the Merlin incubator and powered locker on vehicle ascent. Then on the ISS, our payload was transferred to the Space Tango card for communication. And so we learned just a tremendous amount about logistics and end-to-end verification and integration of these payloads. And we've been building on that since this time for our next launch. We've been focusing on using primary human muscle cells. Certainly countering spaceflight-induced muscle atrophy has been a priority research area from the previous decade. And then, of course, we're looking to those areas now for the next decade. And I'm sure that muscle atrophy will still continue to be a priority area as there's significant decrease in muscle size and force after just about 3 to 6 months in weightlessness. And so as we go forward with deep space exploration, this will be an important area to continue to study. We're also looking at benefiting life on Earth with these systems, enabling this technology, developing disease modeling, and using them to test for drug efficacy and toxicity. So in terms of benefiting life on Earth, the terrestrial problem is age-related muscle decline, a disease that's called sarcopenia. It's a major healthcare issue. Greater than $18 billion a year is spent in healthcare costs. There's no approved therapies. And then, of course, it's a challenge to study age-dependent diseases because they occur over this time frame. So since long-term spaceflight causes this accelerated change and decline in muscle tissue, we can exploit the environment of microgravity to study those mechanisms driving the biology with the mission of understanding the human biology in this environment of microgravity to develop treatments to treat these ailments on Earth. So one of the interesting concepts for our studies is that we are using primary human muscle cells from adults that have been phenotyped. And so we're grouping them into a young age group that's an average of 35 years, an older age group that's 70 years, and the older group is sedentary, while the younger group is an active group. And so there are differences in BMI index, total fat mass, oxygen capacity. And so this is a collaboration with Paul Cohen and Advent Health. And so the volunteers go through 3 visits. On visit 1, they have blood draws and questionnaires. And visit 2, there's a physical test undergone. And then on visit 3 is the biopsy collection. Then we collect the myoblast, convert them into the myotubes from those biopsy cells. And so we have these 2 cohorts, and there's been studies that show that these cells can retain the intrinsic characteristics of their donor. So this is exciting to be able to have these 2 cohorts. So our myobundles are now, we've advanced from our 2D culture and creating 3D myobundles in this microfluidic chip. And so it's a small chip. There's 1 myobundle per chip, and we can form these muscle bundles between 2 PDMS posts, or about 7 millimeter in length. And here this shows a muscle bundle from the older cohort. So we can create, we have platinum electrodes into these chips. We can create an electric field through the chip, and we can simulate that. Here we see a muscle in the upper right panel, a contracting muscle bundle between those posts. And so we can look at the pixel displacement. You see the heat map below. And then from that, get the contraction rate, and then compare the older and the younger cohorts. So if we apply a fairly basic regime of 2 volts, 2 hertz, 30 minutes a day for 5 days, this has been our baseline study, we can see differences, significant differences between the older and younger cohorts. On the top, you see contraction displacement rate for the older cohorts. In the blue line, there's a bit of a waveform, but certainly very similar to background or no contraction really at all. On the lower panel, you see the contraction rate for the younger cohort, and certainly see a larger displacement in the red traces there and a pattern for the contraction rate. If we look at the protein expression, alpha-actin is a contractile protein for sarcomere, which is the contractile function of muscle cells. And so on the bottom, you can see from the young group that we see this number of myobundles. They're starting to fuse into a larger mass. And you see the striatal patterns very distinctly covering these muscle myobundles. Whereas on the top with the older group, you can see some of these striatal patterns, but certainly more diffuse. And the myobundles seem to be less. And so there's quite a distinct difference on a protein level between these two. And of course, we see that functional difference as well. So those were our ground studies. We now put this into the Space Tango payload. We advanced from a 2U system to a 9U system. It could hold 16 of these three-dimensional chips. So a big change and advancement for us in terms of biology. So this payload contains the optics camera system embedded into the top of the payload, so it can run on a rail and take photos. And so the improvement from our payload sense has been a more integrated hosting platform, payload command and receive, on-orbit real-time capture, downlinking of these images and video, and electrical stimulation delivery. And so this is working with Space Tango, so really advancements in autonomous laboratories as well as the biology. We flew this in 2020. Here's the team. We've packaged this into the Space Tango powered locker. And this flew on SpaceX 21 in December of 2020. And so we spent, the payload spent about 9 days in microgravity. We started, once it was plugged in, we could start to capture images. We had 1 day of eSTEM for this experiment. We added the RNA later for preservation and stored it for payload retrieval in January. So on the ISS, we were able to capture images. So on the top left, you see the chip that was pre-flight as we put it into the payload, and then that same chip is shown in the phase contrast image on the upper right, captured with the Cube Lab. And then lower is the actual Cube Lab as it's being transferred to the freezer after we've added the RNA later. It's stored there and then returned to Earth. So on orbit, we had 1 day of eSTEM, so it's not as much of the daily eSTEM, but we did see a movement and contraction in the chip. This just shows the heat map on the left, that there was indeed some contraction from the chip. And then on the right, we see that displacement rate. So less than we've seen on our ground studies, but this was real-time capture on orbit, and that's a significant achievement for our team. We also were able to preserve the cells. We had more than a microgram of RNA per chip, so certainly enough to do transcriptomic studies, and we can compare the gene expression changes between the cohorts in microgravity as well as on ground. This just shows from preliminary data where we've looked at 84 genes that are related to skeletal muscle myogenesis and contractility. And in red, you see those genes that are more highly expressed in the young group versus the old group. So we can start to really investigate differentially expressed genes between the cohorts. So to sum up our autonomous laboratories, past and present, from our NG10 flight in 2018, this was the early development of lab-on-a-chip technology, integrated optics and fluid handling in a pre-programmed experiment. And now we've followed up in 2020. We have a much larger payload. We have integrated communication hosting platform, the on-orbit imaging and video capture, and manual automated performance, as well as an electric stimulation delivery. And so these have been the advancements in these two flights from 2018, 2020. And then moving forward, we have flights for next year planned. We're going to continue studying the contractile function changes and drug effectiveness in these myobundles from the young and the old cohorts on the ISS. We'll be on SpaceX 25 in May of 2022. We'll extend this electrical stimulation time from 1 day to 8 days, perform a longer experiment on the space station to see greater changes. We'll have more daily multiple chip contraction recordings and enhance the autonomous laboratory capabilities. All of that will also be incorporated into our flight after that, which will be in September 2022. So 2 flights next year. Very excited about this. We'll develop drug delivery, evaluate drug dose exposure, and enhance those capabilities as we've been using this MPS system to test drugs and getting some very nice responses. So this is all to develop these disease models of muscle atrophy, to establish drug delivery, and to look for drug efficacy testing. So I think there's some real great uses for these MPS systems in terms of preclinical testing, in terms of looking at natural supplements and human performance. And there's even an agricultural use for these as well in terms of food consumption. So to acknowledge the whole Space Tango team, and MicroGRX is Legrand Mullaney, Austin Hinkle, who've done the chip design at my lab at University of Florida. Shelby Giza did all of the cell biology experiments. Tushar Shenoy is an intelligent undergraduate in our lab. Madalena Parafati has joined and will be working with the upcoming launches. And at the University of Florida Biomedical Engineering Group, we work with Christine Schmidt's lab. Jorge Mojica Santiago has done the simulations and the contraction analysis. And Don Platt at Micro Aerospace Solution has helped with some of the contraction experiments. Paul Cohen, Megan Hoff at Advent Health have collected and pooled these cells that we use. My funding sources at Space Florida, we were part of the 2nd and the 4th call, Cases Organ on a Chip, and NCATS for the Tissue Chips in Space program. Check us out on YouTube. We've got videos from MicroGRX that talk about our preparation for SpaceX 2020, and the University of Florida Communications also has a YouTube video. And I look forward to our panel discussions and any questions that you all have. Thank you.
Thank you, Siobhan. Just a reminder for everybody out there, Siobhan, Dr. Kim, and our last panelists will all be available for a live Q&A at the end of this. So please use the question portal to get your questions in and vote on your favorite questions for those that have already been answered. Next up is Dr. Alan Grodzinski. Dr. Grodzinski is a professor of biological, electrical, and mechanical engineering in the Department of Biological Engineering, Mechanical Engineering, Electrical Engineering, and Computer Science at the Massachusetts Institute of Technology. If there was any doubt out there, MIT loves engineering. Dr. Grodzinski's group focuses on problems motivated by diseases of the musculoskeletal system, including arthritis, connective tissue pathologies, and more generally, the molecular biology and biophysics of the extracellular matrix. As an example, it is well known that traumatic joint injury in humans causes cartilage degeneration and progression to post-traumatic osteoarthritis. But the mechanobiological mechanisms governing cellular transcription, translation, and post-translational responses to physical overload are not well understood. This lack of understanding has limited the clinical therapies that are available for the treatment of these musculoskeletal injuries. This area of research on post-traumatic osteoarthritis was the focus of 2 ISS experiments that Al's team led, and is the subject of the presentation that we will hear now. Al?
Good morning, everyone. It's an honor to be with you all. I'd like to describe our human cartilage bone synovium microphysiological system for osteoarthritis with relevance to drug development and countermeasures on Earth and in space. But first, I'd like to acknowledge our team, in addition to the folks at MIT, our implementation partner TechShot, and investigators at Harvard, Rush University, and Javelin Biotech teamed together with funding from NCATS, of course, as well as CASIS and NASA. I'd like to summarize by first describing some aspects of osteoarthritis and post-traumatic osteoarthritis, then describe our human in vitro microphysiological system for PTOA, and then describe selected results to date on Earth and on station, given the time constraints, which use tissues from donor human knee joints for drug development. First, by way of background, on the upper left, you see the distal femur of a human knee, and it's a very smooth, articulated cartilage surface. On the upper right, unfortunately, is a grade 3 osteoarthritic knee. You can see the tremendous loss of cartilage from the surface. This would be tremendously painful for the patient and would require a joint replacement surgery. It's important to note that osteoarthritis is caused not just by biological factors, but affected by mechanical loading factors as well. OA affects almost 600 million people worldwide now. And in the USA alone, perhaps 65 million people have been cited to have OA, leading to an annual cost of about $480 billion. Importantly, knee OA contributes to that percentage. Over 80% of OA is thought to be knee OA, hence our use of knee tissue for the microphysiologic system. Post-traumatic osteoarthritis is caused by traumatic joint injuries that occur in young and middle-aged healthy people, not just elderly grandma and grandpa. Unfortunately, today there are no disease-modifying drugs for osteoarthritis, only short-acting painkillers that do not halt or even slow the progression of the disease. You can see an example here of a traumatic joint injury. There's initial mechanical damage and immediate inflammation, leading to a high prevalence of post-traumatic osteoarthritis. These are very important patients for clinical trials because we know the instant of initiation of the post-traumatic disease. And immediately after injury, there's an increase in synovial fluid levels of inflammatory cytokines that lead to cartilage degradation. Here, TNF-alpha, IL-1, IL-6 reported in the clinical study, but reported in many other studies as well, a wide range of cytokines. And that leads to the disease. And just to summarize, we start with a normal joint with shiny, gleaming cartilage, bone, ligaments, tendon, other tissues. But after joint injury with initial mechanical impact and inflammation, there's cartilage breakdown, bone remodeling, muscle atrophy, and damage to other tissues. So post-traumatic OA is a multi-tissue joint disease. The only cure right now is joint replacement. And this multi-tissue disease really requires a multi-tissue microphysiological system. Of relevance to our conference here, of course, exercise-related injuries are thought to be the most frequent source of injuries for astronauts living aboard the station. So we need a microphysiological system for drug discovery and countermeasures, and to study effects of joint mechanical loading. And relevant to station and ultimately to deep space travel, disuse versus exercise loading is important to consider. So let me now describe our human in vitro microphysiological system for post-traumatic osteoarthritis. We start with human cadaver knee joints. We have knee joints from over 50 donors so far, male and female, age 28 to 33, Collins grade 0 to 2. Collins grade 0 is perfectly normal knee. Collins grade 1, perhaps a ding or two. Grade 2, some fibrillation, but not osteoarthritis. We purposely exclude osteoarthritic joints from the study because we want to initiate the disease in our own lab and on station, and then see what drugs might be able to mitigate. Of course, I immediately want to thank the donor banks and donor families, without which we would not have been able to perform these studies. So the microphysiological system that we have for post-traumatic osteoarthritis starts with the cadaveric human knee. We then harvest osteochondral plugs from the joint surface. And you can see here a single osteochondral plug on the left. We also harvest synovial joint capsule tissue, and explants from the synovium are then combined with the osteochondral plug to form our cartilage bone synovium, or CBS, co-culture. And that's part of our disease system. We also, however, apply a single impact compressive injury to the cartilage surface, and we use an incubator housed mechanical loading instrument to perform this injury. And this is done because 90% of ACL injuries, for example, are reported to cause trauma to the cartilage surface. So the experimental protocol starts with a cartilage bone plug, and then to simulate the traumatic joint injury and inflammatory response, we start with the injured osteochondral plug, add synovium to the co-culture. That's our disease model with CBS plus mechanical injury. This enables us to study drug treatments, both on Earth and in space. And in the lab on Earth, we've done 2-week experiments, over which time we can collect spent media and then look at the tissue and the media at the end of the study. Let me now describe some of the results on Earth using these human knee tissues. There's ongoing studies of media and tissue in a wide variety of platforms. We look at biochemical outcomes, inflammatory cytokine outcomes, biomarker specific fragment release to the medium, proteomic and metabolomic analyses, and quite a few others. Let me just describe in the limited time here some of the initial results. Question: does the microphysiological system that we have replicate the immediate release of inflammatory cytokines found in human synovial fluid after joint injury and reported in many studies in the literature? So we start with monocultures of cartilage, osteochondral plugs, cultures of synovium, and our disease cartilage bone synovium co-culture. And sure enough, we find that in the synovium alone, or the osteochondral plug co-cultured with synovium, there's dramatic increase in release of inflammatory cytokines, several of them shown here. We use a multiplex panel array. And so the conclusion is that synovium appears to be the primary source of these inflammatory cytokines secreted to the medium, and that's consistent with findings in the literature for human synovial fluids and studies of osteoarthritis. Does our microphysiological system replicate the early release of glycosaminoglycan chains from cartilage seen in early human post-traumatic osteoarthritis? And to explain what I mean, this is a clinical study looking at cartilage changes associated with acute ACL rupture. There were punch biopsies taken from a range of patients, and over the first week or 2 or 3, they found a dramatic loss of a molecule called aggrecan containing little thin glycosaminoglycan chains. That loss is demarked by the loss of staining, toluidine blue staining, and it's one of the earliest events found in human post-traumatic osteoarthritis. Question: do we see such early loss of glycosaminoglycans in our culture system? And the answer is yes. This is results for one donor where over a 2-week period, compared to the control cartilage bone or osteochondral plug, the inflammation condition, cartilage bone synovium co-culture, and the inflammation plus mechanical injury caused significantly increased release of GAG chains into the medium. Now that's one donor. In a study involving 9 donors, we normalized on day 0 to the total tissue GAG content, calling that 1. And then over the 2-week period, we found a significant drop in tissue GAG corresponding to release to the medium in our cartilage bone synovium plus mechanical injury and the inflammation alone compared to controls. So yes, we do see GAG loss in our system. And the forensic evidence is really provided additionally by the loss of this specific proteolytic degradation of the aggrecan molecule, which we can find in the medium using ELISA. And that's very important because it's also found in the synovial fluid of human patients. So the microphysiological system seems to replicate aspects of early post-traumatic osteoarthritis. Can we use it to develop therapeutic drug development, drug treatment on Earth and in space? And first we tried dexamethasone in this system, and we looked at the release of inflammatory cytokines. And again, there's a tremendous increase in the release in our disease condition, the cartilage bone synovium plus mechanical injury. And most importantly and interestingly, dexamethasone at very low dose applied immediately after the model is begun seems to reduce the level of these cytokines pretty dramatically. Now, in consequence, we looked at the ability of dexamethasone applied immediately after injury to reduce GAG loss. And again, these are data from one donor showing rapid GAG loss over a 2-week period in our co-culture plus injury condition, but suppressed down to control levels by dexamethasone. So that's a very promising study. But human donor variability is real, and we're now doing a statistical analysis of all the donor tissues and media that we've been studying in order to come to conclusions. And it may be that because of donor variability, we may require a combination of drugs, not just a single drug, to halt this disease. But our microphysiological system affords the ability to test therapeutics alone or in combination. So finally, let me get to some results on station with relevance to drug development in low Earth orbit and ultimately in deep space. The spaceflight operations and crew procedures are summarized quickly here. We harvest tissue and apply the mechanical injury at our lab at MIT, ship to Kennedy Space Center, and then following launch and travel to station and docking, we begin the experiment. It's a 3-week experiment where we collect media, and at the end, we can take the media and on return to Earth, examine the media as well as the tissue. And to do this, our implementation partner, TechShot, helped us to design and fabricate tissue cards into which we can place osteochondral plugs. And in some of the cards, to duplicate the disease state, we also suture the synovium explants. So we have the co-culture occurring on this card. The cards are placed into a specially designed culture chamber that TechShot designed and developed. The culture chambers are then put into TechShot's modules along with sample bags and nutrient bags. The bags in the module can then implement nutrient media transfer into the culture chamber every 2 days to try to simulate what we do on Earth. And we were lucky enough to get 2 donors for the most recent launch on SpaceX 21. The modules are then put into the carousels on TechShot's instrumentation on station. And just to describe some initial results, ongoing studies, of course, are now in place. We did find that the inflammatory profile, cytokine profile that we saw in flight showed trends similar to that in the microphysiological system on Earth. That is, they were highest in the disease state. The black dots are our cartilage bone synovium co-culture plus mechanical injury. But dexamethasone in this donor was able to reduce those levels significantly. I don't have time to show the data, but we also found gag release profiles on station mimicked the trends that we see on Earth. But again, we have to worry about donor variability or human variability, and those studies are underway. There's still a lot of analyses to go, and I will be thrilled to report those when we're done. Let me now, in summary, just offer some conclusions and further thoughts. Our microphysiological system appears to replicate certain critical events in the initiation and early progression of post-traumatic osteoarthritis found in human subjects after joint injuries on Earth. Analyses to date from station suggest that our microphysiological system also demonstrates changes in human knee tissues indicative of the earliest events in the initiation and progression of post-traumatic osteoarthritis. The use of such microphysiological systems, we believe, on LEO-based platforms could enable further accelerated disease modeling and provide unique insights into disease progression and development of therapeutic interventions. The example so far is low-dose dexamethasone, which appears to be, to us, a potential disease-modifying drug if applied at the appropriate time early enough and at the appropriate dose, having effects on stations similar to those on Earth so far. In microgravity, humans experience musculoskeletal deterioration, and extended spaceflight can alter joint integrity in a fashion relevant to progression of OA. The challenge here is that post-traumatic osteoarthritis can take 10 to 15 years to develop, and we need slightly longer experimental times on station than the 3 weeks that we have now had in order to look at the intermediate stages of disease. The effects of altered loading on joint cartilage in space may affect the rate of cartilage breakdown leading to OA, and microgravity uniquely enables studies of joint disuse versus exercise in the management of OA and post-traumatic OA. Now the challenge for us is to develop instrumentation for loading the microphysiologic system on station. And we've been doing just that. We've started studies in which we can use an incubator housed system to apply mechanical loads to the osteochondral plugs in a specialized chamber that allows co-culture with synovium. And we're hoping that these experiments will be relevant to exercise protocols currently used on station. And with that, I'd again like to acknowledge our coworkers and different members of the team, and thank you very much for your attention. We hope to continue the discussion in the session.
Thank you, Al. I'm now joined by each of our panelists who video presentations you were just able to see. We've been receiving questions over the Q&A line, and some of those have received quite a number of votes from the audience. So I'll begin with the first one, and this is for all of the panelists here, and I'd like to begin in order, so we'll start with you, Ducko, and then go to Siobhan, and then to Al. Space is hard, science is hard. How is the data generated from your research and the technology development required for performing your research on the ISS being translated into future clinical applications on Earth and in space? Ducko?
Yeah, thank you for the question. So our research work is still in the discovery phase, and it's relatively far from the clinical use. Yeah, of course, we aim to determine the pathway involving the spaceflight-induced cardiac dysfunction and identify the potential therapeutics, which may be utilized clinically in the future. We are also hoping that our information and identified therapeutics will translate to the treatment of the age-related cardiomyopathy.
Thank you, Dr. Siobhan.
Yes, great question. So we're using these donor-specific cells, and the goal is to be able to use these for preclinical testing and evaluation. And in using the different cell-specific cell populations, we're asking really on one level how these cells perform in microgravity, and can we understand the mechanisms driving the biological change, and does that manifest into a disease that mimics this age-related atrophy? But then on another level, since we have these two cell cohorts, we can look at the sensitivity of each one of those, is the magnitude of change different between the younger cohort and the older cohort, for example? And then comparatively, how do they compare with each other? Is there convergence or divergence in their change? And so both on microgravity and ground. So I think that's really going to feed into disease modeling and preclinical model that we can use for looking at therapeutic intervention. And then on the technology side, we've been heavily investing into the cube lab science, and the idea there is that it's very challenging to be in a closed system and an automated system. If you think of the iterative experiments you have to do on the ground, this is one box. But the idea, once all of that processes, standard protocols are operating in the cube lab, I foresee that we could put this in either an incubator on the ground for 6 months or a year and look at long-term chronic evaluation, as well as putting these in the locker system on the space station for long periods of time. These miniaturized tissue chips are robust in culture. There's low volume needed to feed these cells. And so then you could do multiple twin studies, for example. So this is also going to be very important as preclinical technology in the future.
Thanks, Siobhan. Al, you spoke to this a little bit in your presentation about inter-patient variability and the need to develop high-throughput systems such as this. So specifically, how is the adaptation of your experiment from your vast experience with ground-based microphysiological systems and technology development for space, how has that pointed you possibly in new directions for improving your model system?
Yeah, thanks, Mike. It's a very, very important question. I think for our system, the most interesting and important thing we're finding right now is the potential for direct testing of therapeutics. It seems like our microphysiological system can, certainly on Earth, and we're finding initial results on space as well, emulate the initiation and progression of an osteoarthritic-like disease. And that enables us to test whether appropriate use of therapeutics at the right time and at the right dose, which can be a function of human donor variability, or we're all different from each other on this screen, and we may not need the same dose, but the timing is very important, when to apply a drug. Do we need a combination of therapeutics? Is there going to be one magic bullet to cure osteoarthritis? So far, there are none, but it's probably true that because of human variability, we might be able to use a combination of therapeutics. So these are things that we're directly testing right now.
Thanks, Al. We have under 2 minutes left, so I'm going to ask you to keep your answers to this question in under 15 seconds. So I know that's not fair, and I'd like to begin with Siobhan. So Siobhan, would you comment on the suitability? You mentioned a CubeSat in your previous answer. So do you think it would be possible to further automate these systems, these microphysiological systems, for use in a CubeSat platform without crew support?
Right, and that's been our big focus in our last 2 launches, is perfecting the mechanics. And as you know, in a closed system, if you change temperature, you change pressure, everything's got to work harmoniously. And so this has been a big investment for the ability to take these out long term, and which is going to be important for deep space exploration, to see, you've got a young cohort that's like an astronaut and the other one that might be more like an average person, to see the effects long term. And so and there's no astronaut intervention, but also with these CubeSats, there's the ability to do manual commands to perhaps fix things, remove a bubble, move a camera system. And so this is all very important.
Very good. Al, same question. Do you believe you could automate your system for use in an untended CubeSat format?
Yeah, we really hope that's possible. We've had 2 flights and have had to iterate the technology as we go. And so I think as a result of the knowledge we've gained, we'd be able to get to automation. That's the hope.
Ducko, same question for you with your heart construct.
I see no reason CubeSat would not work for our future experiment. However, we need to investigate this further.
I'd like to take this opportunity again to thank each of the panelists today. This has been an excellent and very informative session for us. We at the ISS National Lab and working with our partners at NASA, each of the implementation partners who've worked with the science teams here, and especially without the leadership and guidance again of the National Institutes of Health in not only funding but taking the lead and keeping this important technology development on the forefront of our minds, we would not have been able to achieve the great success that you've each brought forward today. So I thank each of you for your participation today. I thank the audience for their interest, and I will kick it back over to Jim.
Excellent. Thank you, Mike. And Doctors Milani, Grodzinski, and Kim, thank you so much. Amazing stuff that you're all doing and that you're continuing to do. We appreciate that session. That was great. So now, yesterday, we honored pioneers in ISS research and development for both 2020 and 2021, because so much good research and innovation has and is taking place. We're going to recognize even more of these incredible people and their work. So let's take a look at this video highlighting today's award winners.
I'm Kurt Costello, the ISS Chief Scientist, and today we're going to continue our ISSRDC awards. Yesterday you heard about our first 6 candidates in the categories of compelling results and innovation on board the ISS. Today we'll hear about those next 6, so let's get started.
Our first award is for a compelling result in the fields of biology and medicine and goes to Dr. Se-Jin Lee. Sponsored by the ISS National Laboratory, the Mighty Mice in Space investigation, conducted in 2020, used rodent models to investigate the potential benefits of targeting the dual myostatin and activin signaling pathways with a unique therapeutic to prevent both skeletal muscle and bone loss due to disuse. The study, also led by his wife Dr. Emily Germain Lee, was a product of decades of work to understand both bone and muscle loss. Results of this study will be particularly relevant for conditions characterized by muscle loss due to disuse. This could include patients recovering from hip fracture surgery, intensive care patients, and the elderly. Applications extend to a wide range of clinical settings in which debilitating muscle loss occurs, including muscle degenerative diseases like muscular dystrophy and muscle wasting. It also has implications for improving bone health, such as in patients with osteoporosis, and may provide the basis for pursuing strategies to protect against muscle and/or bone loss during spaceflight. Our next award for innovation in commercialization and non-government utilization goes to Dr. Gene Boland of TechShot Inc. In September 2019, TechShot and their partners at InScript successfully printed and cultured human cardiac cells in their biofabrication facility, or BFF, on the space station. This success represents the first step for a potentially game-changing technology. The 3D printing of soft biological tissues on Earth is limited by tissue collapse due to gravitational forces. In the microgravity environment of the space station, researchers can print tissues and potentially organs free from the constraints of scaffolding or structural support. TechShot has come a long way in developing the BFF. After designing the hardware, it was tested on multiple parabolic and suborbital flights. Specialty inks consisting of adult human cells, such as stem or pluripotent cells, and adult tissue-derived proteins for printing in microgravity had to be created. After printing, the tissues were cultured to strengthen over time in order to become self-supporting tissues capable of withstanding the rigors of Earth's gravity. This successful demonstration opens the door to new soft biological materials and complex architecture not possible for 3D printing on Earth. Our next award for innovation in technology development and demonstration goes to the team behind the Small Optical Link for the International Space Station, or SOLAS. SOLAS is a joint project based on fundamental research conducted by the Sony Computer Science Laboratories and the JAXA Space Exploration Innovation Hub Center to demonstrate in-orbit and long-distance laser communication technology. SOLAS uses optical disc technology that Sony has employed in household devices such as CD players and is a good example of the public-private partnerships as well as commercial use of the ISS. This successful demonstration enables real-time mass data communications for future inter-satellite communications and communications with ground stations. It also raises expectations for high-rate internet connections for regions in the world that do not yet have ground-based internet access. Furthermore, this technology could enable wide-range human space exploration missions to the Moon and Mars. The SOLAS team is made up of Iwamoto Kyohei and Komatsu Hiromitsu of Sony Computer Science Laboratories, Sawada Hirotaka and Koda Daike of JAXA's Space Exploration Innovation Hub Center, and JAXA's Ikeda Toshitame of the GEM Kibo Mission and Integration Center. Our next award is for compelling results in biology and medicine and goes to Afshin Beheshti, PhD of KBR, NASA Ames, for leading the most comprehensive study of the effects of spaceflight on living organisms to date. Using biomedical profiles from 59 astronauts and data from NASA's Gene Lab and NASA's Twin study, his team used a multi-omic systems biology approach, uncovering insights into the fundamental biological mechanisms affected by spaceflight across organisms, resulting in 3 discoveries. First, spaceflight effects were more evident in isolated cells than whole organs, suggesting that tissue complexity plays an essential role in response to space-related stress. Second, the liver undergoes more differential gene and protein expression changes than other organs, consistent with the role the liver plays as a dynamic and critical hub in sensing changes in blood composition and maintaining homeostasis. Third, spaceflight impacts mitochondrial function at the genetic, protein, and metabolite levels of cellular, tissue, and organismal biology, indicating mitochondrial stress as a consistent phenotype of spaceflight. For our next award, we have another compelling result in biology and medicine, which goes to Dr. Masayuki Yamamoto and the MHU-3 mission team. Professor Yamamoto and his team focused on the role of environmental stress-responsive transcription factor, NRF2, and revealed that the stresses of spaceflight presented as aging-like changes of plasma metabolites, and that NRF2 is important for maintaining homeostasis. In addition, these metabolic changes in space were compared with large-scale human metabolome data accumulated by the Tohoku Medical Megabank, TOMO, project in Japan. Metabolites in plasma from young adults aged 20 to 40 years old and older, aged 60 to 80 years old, participants in TOMO cohort data showed good accord with the space mouse results. Several new capabilities were demonstrated in this experiment, including 100% live return of genetic knockout mice after 31 days of spaceflight for the first time, a new blood collecting device, and procedure enabling continuous pre-launch, in-flight, and post-landing blood collections, and onboard monitoring of individual mouse condition, including food intake and water consumption. In addition, this study initiated a data archiving activity across the entire JAXA rodent mission. The Integrated Biobank for Space Life Sciences was developed as a platform to enhance data and sample sharing related to space biology. Our final award for compelling results in biology and medicine goes to Dr. Rasha Hamameha, Director of Medical Readiness Systems Biology at Walter Reed Army Institute of Research. Rasha has flown multiple investigations to the space station to better understand the effects of spaceflight on tissue and wound healing. Her research program, sponsored by the Department of Defense, Space Test Program, Walter Reed Army Institute of Research, and the ISS National Laboratory, consists of investigations that have steadily built upon each other and increased in complexity. Microgravity has been shown to impair healing. Rasha's work to identify the molecular foundations of wound healing that are vulnerable to spaceflight-induced stress may reveal biologically relevant pathways for the next generation of wound healing therapies. The results from these projects will have implications for tissue and bone regeneration, both in spaceflight and on Earth.
And congratulations to all of our award winners. Fantastic work by all of you. Very well done. We're going to take a short break right now, but be sure to come back right at 12:30 for our industry spotlight with NanoRacks CEO, Jeff Manber. We'll see you all then.